somewhere inside your body, right now, a tiny mistake is being copied into a new cell. It happened before you were born. Nobody warned your parents. Nobody could have. Scientists just found the exact spot where this mistake keeps happening, over and over, in almost every human being on Earth. They are calling it a danger zone, and what they found inside it might explain why certain diseases run in families that seemingly came out of nowhere. For decades, we thought we understood where mutations come from. Sunlight damages skin cells. Chemicals in cigarettes damage lung cells. Copying errors happen every time a cell divides. These were the villains scientists blame for most genetic diseases. But there was a fourth source hiding in plain sight, right at the very beginning of thousands of genes, and almost nobody was looking there. That is, until now. A team of researchers based in Barcelona, working with genetic data from hundreds of thousands of people, just published a study that flips a piece of basic biology on its head. Their paper appeared in Nature Communications, one of the most respected science journals in the world. And the headline finding is almost hard to believe. The very start of a gene, the exact spot where your cells begin reading DNA instructions, is far more likely to mutate than anywhere else nearby. Not by a small margin either. The first 100 letters of genetic code right after a gene begins are 35% more likely to carry a new mutation than random chance would predict. Let that sink in for a second. 35% higher risk, concentrated in one tiny stretch of your genome, repeated across thousands of genes in every single human being alive. This is not a rare glitch. This is a built-in feature of how your cells work, and it has been quietly shaping human health since before recorded history. To understand why this matters, you need to picture what a gene actually is. Think of your DNA as an enormous instruction manual, written in a 4-letter code, stretched out inside every cell in your body. Genes are individual chapters in that manual. Each one tells your cells how to build a specific protein, whether that protein helps grow bone, fight infection, or regulate your mood. But before a cell can read a chapter, it has to find the exact starting page. That starting page is called the transcription start site. It is the spot where a molecular machine locks onto the DNA and begins copying the instructions into a working script called RNA. Researchers have now discovered that this starting page is a chaotic place. It pauses. It sometimes restarts. In some cases, it even fires in the wrong direction entirely, reading a few letters backward before correcting itself. While all of this stopping and starting is happening, tiny sections of the DNA double strand come apart and briefly expose a single strand to the environment inside the cell. An exposed single strand of DNA is fragile. It is far easier to damage than DNA that is safely wound up in its usual double helix shape. And damage, when it is not repaired perfectly, becomes a mutation. This is the mechanism behind the danger zone. It is not one specific toxin or one specific ray of sunlight causing the damage. It is the ordinary, everyday process of a cell simply trying to read its own genes. The machinery meant to bring genes to life is, in a strange twist, also the thing most likely to scar them. Here is where the story gets even more important. The researchers found that a huge portion of these mutations do not happen randomly throughout your lifetime. They happen incredibly early, in the first few rounds of cell division right after conception. Before you were even the size of a poppy seed, your cells were dividing at an astonishing pace to build an entire human body from a single fertilized egg. A speed like that comes with a cost. Cells under that kind of pressure to grow fast do not always have time to fix every tiny mistake in their DNA before passing it along to the next generation of cells inside the growing embryo. The result is something scientists call a mosaic mutation. This is different from the mutations most people picture when they think about genetics. A mosaic mutation does not exist in every single cell of your body. It exists in a patchwork. Maybe it is present in a portion of your skin cells, but absent from your blood. Maybe it shows up in some brain tissue, but not in your liver. This patchy, uneven distribution is exactly why these mutations have been so difficult to detect using standard genetic testing. Now here is the part that should genuinely concern you in a productive way. A parent can carry one of these mosaic mutations without ever knowing it. It might be sitting quietly in a tissue that never causes a symptom, tucked away where it does no visible harm to that parent at all. But if that same mutation happens to be present in the eggs or sperm that parent produces, everything changes for their child. When a mosaic mutation is passed down through reproduction, the child does not inherit a patchwork version. The child inherits that mutation in every single cell of their body because it was there from the very first cell that formed them. A mutation that was silent and harmless in the parent can become a fully expressed disease-causing mutation in the next generation. This is a genuinely new piece of the puzzle for understanding conditions that seem to appear out of nowhere. Families sometimes see a child develop a serious condition with no obvious history of it in the family. Doctors have long assumed these cases come from spontaneous, totally random mutations that could strike anyone at any time. This new research suggests something more specific is often going on. Some of these apparently random mutations are not random at all. They are landing, again and again, inside this newly identified danger zone, a stretch of the genome that turns out to be one of the most mutation-prone regions we have ever measured. To reach this conclusion, the scientists did not rely on a small sample. They analyzed genetic information from 150,000 genomes collected through the UK Biobank, one of the largest genetic databases on the planet. They then cross-checked their findings against another 75,000 genomes from the Genome Aggregation Database, often called GNOME-AD by researchers, which pools sequencing data from labs around the world. On top of that, they layered in detailed findings from 11 separate family studies that specifically tracked mosaic mutations across parents and their children. That is an enormous, carefully verified data set, and the pattern held up consistently across all of it. When the researchers looked closely at which genes were most affected by this mutation hotspot, 3 categories stood out again and again. Genes involved in cancer, genes involved in brain activity, and genes involved in limb development. Think about what that actually means. The exact genes most likely to be hit by these newly discovered mutations are tied to some of the most serious and life-altering conditions a person can face. But there is a twist in the data that actually offers a strange kind of reassurance. When the researchers looked at extremely rare genetic variants, the kind that tend to be very recent mutations, they found a strong excess sitting right in this danger zone, exactly as expected. However, when they looked at older mutations that had become more common across the population over many generations, that excess mostly disappeared. In plain terms, that means natural selection is actively filtering many of these harmful mutations out over time. Families carrying dangerous mutations in these regions, especially the ones tied to cancer and brain function, are statistically less likely to successfully pass those mutations on to future generations. Evolution, slowly and imperfectly, is doing some cleanup. Still, that cleanup process is not instant, and it is not perfect. It happens across many generations, which means individual families today can absolutely be affected before that filtering ever has a chance to work. Understanding exactly where these mutations are most likely to appear gives researchers and doctors a genuine head start, instead of searching blindly across the entire genome for an explanation. This discovery does something else that might sound technical at first, but is actually enormously practical for medicine. Genetic researchers use something called a mutational model. Think of it as a baseline expectation, a prediction of how many mutations should naturally show up in a given stretch of DNA if nothing unusual is happening. Doctors and scientists compare real-world genetic data against that baseline to figure out which mutations are worth worrying about and which ones are just normal background noise. The problem is that, until now, none of these models accounted for the danger zone. If a model does not know that the start of a gene is naturally mutation-rich, it can badly misjudge what it is looking at. Imagine a model expects to see around 10 mutations in a certain region, based on old assumptions, but actually observes 50. That looks alarming at first glance. It looks like something dangerous is happening in that gene, but if the true, corrected baseline for that region is actually 80 mutations, then 50 is not an alarming excess at all. It is actually fewer than expected, which is a sign that natural selection has been quietly removing harmful versions of that gene from the population. Without the corrected baseline, a scientist could completely misread the signal and miss the true importance of that gene entirely. It is actually fewer than expected, which is a sign that natural selection has been quietly removing harmful versions of that gene from the population. Without the corrected baseline, a scientist could completely misread the signal and miss the true importance of that gene entirely. This is exactly why the discovery of the danger zone is not just an interesting fact about biology. It forces a recalibration of tools that are already being used in real hospitals and real research labs right now to interpret real patients' genetic results. Every mutational model that ignores this hotspot has been working with a baseline that was quietly wrong. There is a second major consequence buried in this research, and it involves how genetic studies are typically designed. A huge number of genetic disease studies work by comparing a child's DNA to both of their parents' DNA. If a mutation shows up in the child but is completely absent in both parents, researchers flag it as a new, spontaneous mutation worth investigating. This method works reasonably well for mutations present in 100% of a person's cells, but it fails, quietly and consistently, for mosaic mutations. Remember, a mosaic mutation might only be present in a fraction of a parent's cells, sometimes too small a fraction for a standard blood test to ever catch it. If a parent's mosaic mutation does not show up in the blood sample used for testing, but that parent still passed it on through their eggs or sperm, the child's version of that mutation will incorrectly look like a brand new spontaneous event. In reality, it was hiding in the parent all along. This means an unknown number of genetic studies conducted over the past several years have been quietly filtering out and discarding. Exactly the kind of information that could help explain the origins of certain inherited conditions. The researchers behind this study offered a practical fix for that blind spot going forward. One option is to look for patterns of mutations occurring together, since co-occurring mutations can be a signature clue that a mosaic mutation is present in a parent, even if it was not obvious the first time around. Another option is going back through old, previously discarded genetic data, specifically revisiting mutations that occurred near the transcription start sites of the genes most affected by this hotspot. In other words, some of the answers scientists have been searching for may already be sitting in old datasets, simply mislabeled or overlooked the first time around. Stepping back, the significance of this discovery goes well beyond one single study. Since the human genome was first mapped over 2 decades ago, scientists have known about the 2 classic sources of mutation: random errors during DNA replication, and damage from external forces like ultraviolet light or certain chemicals. Finding a brand new, distinct source of mutation, especially one that specifically affects the part of DNA passed down to future generations, is genuinely rare. It does not happen very often in modern genetics, and when it does, it tends to ripple outward into dozens of other areas of research. If you have ever wondered why some serious conditions seem to appear in families with absolutely no prior history, this research offers one compelling piece of the explanation. Not the entire explanation, because human disease is almost always shaped by a mix of genetics, environment, and pure chance. But a real, measurable, and previously invisible piece of it, hiding in one of the most functionally important regions of the entire human genome. If this kind of deep, mind-bending science content is exactly what keeps you coming back, do me a quick favor and hit that like button right now. It genuinely helps this channel keep making videos like this one. What makes this discovery even more remarkable is the timing of it. We are living in an era where genetic sequencing has become faster, cheaper, and more accessible than at any point in human history. Millions of people have already had their genomes sequenced, whether through medical testing, research studies, or consumer genetic kits. And yet this particular pattern, sitting right at the start of thousands of genes, went unnoticed until massive datasets like the UK Biobank became available for this kind of large-scale analysis. It took data from hundreds of thousands of people, combined with careful statistical modeling, to finally reveal a pattern that had been there the entire time, invisible simply because nobody had ever looked at the data from quite this angle before. That is often how major discoveries work in modern genetics. It is rarely a single dramatic breakthrough moment. It is usually the slow accumulation of enormous datasets, followed by someone finally asking the right question of that data. In this case, the right question was surprisingly simple. Are mutations evenly distributed across the genome, or are certain locations more vulnerable than others? The answer turned out to be no, they are not evenly distributed. The difference is large enough to matter for real patients.